Hi again, everybody. Today we're going to talk about neurotransmitters. So we left off talking about neurotransmitters entering the synaptic cleft and then triggering some type of response in a target cell. Today I'm going to more formally introduce those neurotransmitters and talk about their structure and their function. And then we'll finish up with how the function of neurotransmitters can actually be altered via neuromodulation. All right, let's go. Worksheet 1.6 has a couple tables on them, so you'll want to fill that out when you're prompted to do so. The objectives for today are going to be to first look at the classification of neurotransmitters. So there's 4 classes that are based on structure, and then we can look at functional classifications and then some broad actions of these. And my logic with this is if you can understand how they're classified, it's going to help you understand the functionality and their action, which is going to be important for when we move into the peripheral nervous system. We'll then look at features of neurotransmitters, and we're going to focus on acetylcholine as kind of like our model neurotransmitter. Then we'll move into neuromodulation, where we'll talk about specifically the role of endocannabinoids, but we'll talk about all of them in general. And then we'll move and finish with antidepressants and your mental health. Science comics for today. Got neurotransmitter related content here. So my brain sees a dopamine. My brain releases dopamine. Nice pun on dopamine there. And then who can forget the Swift West showdown at the MTV Music Awards? So we'll see, is dopamine the greatest neurotransmitter of all time? And then just some general college-related humor here. This is one of my favorites that I think I've shown every year since I've been here. It says, terrible professor and overall terrible person. So this is obviously I rate my professor. Had to drop the class because she doesn't give any grades besides tests and in-class pop quizzes. Also, she is not a good person. I saw her at Chipotle, and she filled her water cup up with iced tea after complaining to the manager. Clearly not a good person or teacher. All right. There are currently thought to be around 100 known neurotransmitters, and these are all small organic molecules. So organic molecules, remember, have some type of carbon frame, whether that's ring structure or chain, and then they're associated with things like hydrogen, oxygen, and nitrogen oftentimes. And neurotransmitters can have inhibitory or excitatory effects. So if we think of a few, we have adrenaline here, which we call epinephrine, and that's associated with our fight or flight response. We have serotonin. We think of that as the mood neurotransmitter. And then we have endorphins. Endorphins, we associate it with that post-workout high where we're just feeling euphoric. So let's take a look.
This is such a great question. The thing is, it's not exclusive to running. You can get a high from a really good workout. And this goes back to our days as hunter-gatherers when we used to perform what's called persistence hunting. It's where you would run mile after mile after mile to hunt the prey down. But that running causes inflammation, which can push on nerves, and that can cause pain. So your body started to secrete these hormones designed to numb the pain and also make you euphoric. You started to like it, so you would do it more. We call those endorphins. There's other examples of those in the body other than endorphins, but an endorphin, it literally means endogenous morphine. It binds to the same receptors that opiates do, which is why you get high from it. The user PGT8 nailed it. Awesome job, PGT. Now, today's question comes from Desi538, and Desi wants to know, why do our eyebrows and eyelashes not grow as long as the hair on the top of our heads?
Let's run through some general characteristics of neurotransmitters then. The first is going to be that they are synthesized by neurons and then stored within vesicles within the synaptic knobs. So pop quiz. What type of axonal transport is this? All right, that's right. It is anterograde fast axonal transport. So we move toward the axon terminals there where we're going to store those vesicles. So that's going to involve that motor protein kinesin we talked about in the last lecture. Secondly, neurotransmitter is going to be released from the vesicles when an action potential triggers calcium entry into that synaptic knob. So we can see on this image here, there's going to be a calcium influx, allowing that vesicle to fuse and dump that neurotransmitter content into the synaptic cleft. Third, that neurotransmitter is going to bind to a receptor in a target cell. So this could be a postsynaptic neuron, it could be a muscle or a gland. All right. And then finally, that binding of the neurotransmitter is going to trigger some type of physiologic response within that target cell. So for example, here if we're talking about acetylcholine, that is going to help lead to a skeletal muscle contraction. First, we'll start. We can classify neurotransmitters in a few different ways. So the first is just based on their chemical structure. If we do it this way, there's just 4 groups. You have your acetylcholine, it's got its unique structure. Then we have our biogenic amines, our amino acids, and our neuropeptides. Then we can also classify based on function, and we can divide this into the effects of that neurotransmitter versus the action of the neurotransmitter. If we're looking at effects, it can either be excitatory or inhibitory. Excitatory meaning it causes an excitatory postsynaptic potential, inhibitory postsynaptic potential. Actions, they can either act directly, where binding of neurotransmitter is going to directly open some type of ion channel, or indirectly, we're going to use some type of secondary messenger. Let's begin with chemical structure. So as mentioned, there's 4 categories based on structure. And for example, these are the biogenic amines that I'm showing here. So they all have this amine group. So let's start with acetylcholine. Acetylcholine is unique from all the others, so it gets its own category. You'll notice right off the bat that we don't see any ring structures. It's not a peptide, so it's not a string of amino acids, and it's not an amino acid itself. So this is how we can recognize acetylcholine. Next up, we have the biogenic amines. So these are derived from certain amino acids by removal of a carboxyl group. So looking at our amino acid down here at the bottom, we can cleave off our carboxyl group. We're using that enzyme decarboxylase, and we are left with some type of biogenic amine. But this is going to depend on this functional group here. This is how we differentiate them. So let's take a look at the different functional groups. Broad interest. So if that functional group contains a catechol group, then it is a catecholamine. So this is a subclass of biogenic amine. If that functional group contains an indole group, it's an indolamine, the other subclass of biogenic amines. So biogenic amines are one of 2 things. They're either catecholamines or indolamines, and it depends on that functional group. Okay, the first type of biogenic amine we'll cover are the catecholamines. So catecholamines are synthesized from the tyrosine amino acid. So it's a modified tyrosine amino acid. And what we'll look for then in these final neurotransmitter products are they're containing that catechol group. So we see this catechol group within all of these neurotransmitters. And they include some of our more common neurotransmitters, epinephrine, norepinephrine, and dopamine. So made from modified tyrosine. The CNS. And our other class of biogenic amines are the indolamines. Okay, and these include histamine, synthesized from histidine, and then serotonin, which is synthesized from tryptophan. So histamine is modified histidine, serotonin is modified tryptophan. And again, these are all going to contain an indole group. Less obvious within the histamine, it's kind of a broken ring structure there, but then very obvious within the serotonin. Then we have our amino acid neurotransmitters, and these include glutamate, glycine, serine, and GABA, or GABA-aminobutyric acid. Okay. So your amino acids are going to have, you can think back to 101, the structure where you're going to have amino group, a hydrogen, an R group, which is some type of variant, and then your carboxyl group. And remember, that carboxyl group is what we just cleaved to make our biogenic amines. All right, and that brings us to our last structural category here, the neuropeptides. So as you can probably guess by that name, peptides, which are strings of amino acids. They can be 240 amino acids long. They include things like endorphins, enkephalin, cholecystokinin, and substance P. So I'm pointing out substance P, and that P, kind of a weird name. That P actually stands for powder or preparation. When it was first discovered, they named it that. But I remember it because P and pain, and substance P is involved in pain reception. But anyway, notice that substance P is categorized as excitatory. So that's going to move us into our next classification. We can also classify by function. All right, so let's turn to function, and let's start by looking at classification of function by looking at the effect of that neurotransmitter. When classifying neurotransmitters by effect, we look at whether they are excitatory, which can help lead to a depolarization and action potential, or whether they are inhibitory, and they lead to hyperpolarization, moving membrane potential further from threshold. Some neurotransmitters are going to open chemically gated cation channels. So we can see sodium and potassium gates here. So neurotransmitters are going to fuse across that cleft, bind and open those channels. Okay, so what we're going to see, the overall effect then, sodium is going to rush in to that cell. A little bit of potassium is going to leave. And this is going to be excitatory because with that influx of the positively charged sodium, this is going to be EPSP, excitatory postsynaptic potential. Okay, so that cell is going to become less negative, can lead to depolarization and action potential. Now, some neurotransmitters are going to open chemically gated chloride channels or potassium channels. And with this instance, we'll see that the cell becomes hyperpolarized or more negative. So let's say we have chloride influx with the opening of that chloride channel into the next cell. It's going to become more negative, and that is going to create a hyperpolarization within that cell, and it's going to be inhibitory, okay? IPSPs. And I want to point out here that both excitatory and inhibitory, so EPSPs and IPSPs, can be generated because postsynaptic neurons are capable of binding many different neurotransmitters. So we can get EPSPs from some, they're excitatory, or IPSPs from others, they're inhibitory. And it's only when we overcome the IPSPs or we get enough EPSPs to hit threshold that we'll get action potential. Human anatomy. All right, and finally, classification by action. Actions of neurotransmitters can either be direct or indirect. So here we're looking at a direct neurotransmitter action. We see the neurotransmitter comes in and it's going to bind to this ion channel. Binding to the ion channel opens that channel and allows for ion exchange between the ECF and the cytosol. Now we can see an example of indirect action. So indirect action here, we see that the neurotransmitter binds a receptor, and then binding of that receptor is going to activate a second messenger pathway, and that usually involves a G protein. So we've talked about G proteins before. And then ultimately, what's kind of cool is the second messenger ultimately can lead to many more downstream diverse effects. This can be opening ion channels, or it can be activation of some enzymatic pathway, transcribing some genes for synthesis of new proteins. There's lots of options here. Now we're going to investigate some broad actions of some of our neurotransmitters. All right, so you want to turn to your worksheet now and fill out the table while we watch this short bit of this Khan Academy video on just some brief descriptions of some of these neurotransmitters that we've covered. We'll talk about their functions within the body.
Types of neurotransmitters. So now I'm going to introduce some important neurotransmitters in these different groups, and I'm going to mention some of their functions. But don't worry too much about their functions right now, because they do so many different things in different parts of the nervous system that we'll come back to all of that in other videos. But I just want to briefly introduce the different important neurotransmitters in each of these classes. Starting with the amino acids, important amino acid neurotransmitters are this one, which is called glutamate. This one, which is called gamma aminobutyric acid, which pretty much everybody just shortens to GABA, G-A-B-A, for gamma-aminobutyric acid. And then this one, which is glycine. Glycine. Now, glutamate is the most common excitatory neurotransmitter of the nervous system. So let me just draw a big plus sign above glutamate here, because most of the time in the nervous system, when a neuron is releasing a neurotransmitter that is exciting its target cell, most of the time that neurotransmitter is glutamate, because it usually causes depolarization of target cells so that it excites them. GABA and glycine are the most common inhibitory neurotransmitters of the nervous system. So let me just write some big minus signs above GABA and glycine, because they usually cause hyperpolarization of target cells and inhibit those target cells. GABA is the most common inhibitory neurotransmitter in the brain, while glycine is the most common inhibitory neurotransmitter in the spinal cord. So that the amino acid neurotransmitters are really involved in most functions of the nervous system. Pretty much if you think of anything the nervous system is doing, at some point in the chains and networks of neurons, glutamate, GABA, and or glycine are probably involved in moving information through those networks. There are many important monoamine
And when he says monoamines here, he's talking about those biogenic amines, the ones that are derived from the amino acids. There's modified amino acids.
Transmitters. I'm just going to mention these 5 that are arguably the most important. The first one here is serotonin. Serotonin. The next one here is histamine. Histamine. The next one is called dopamine. Dopamine. Then this one is epinephrine. Epinephrine. And right next to epinephrine is its close cousin, norepinephrine. Norepinephrine. So that all 5 of these are monoamine neurotransmitters, but these 3, dopamine, epinephrine, and norepinephrine, are also called catecholamines. The monoamines play a lot of different functions in the nervous system, and in particular a lot of functions of the brain, including big things like consciousness and attention, and cognition, or thinking, and emotion, or us having feelings. Norepinephrine is also released by some autonomic neurons in the peripheral nervous system. Many disorders of the nervous system involve abnormalities of these monoamine neurotransmitter systems, and many drugs that people commonly take affect the monoamine neurotransmitters. There are many important peptide neurotransmitters, including a group of peptide neurotransmitters called the opioids. Opioids. And the opioids are a group within the bigger group of the peptide neurotransmitters. And this one is one example of an opioid. This is endorphin. Endorphin. And the peptide neurotransmitters play a role in many functions of the nervous system, but the opioids in particular play a big role in our perception of pain. And a number of pain medications affect the opioid neurotransmitters. And last, but definitely not least, are the other neurotransmitters. Now, usually when there's an other category of anything, that means it's not very important. But in the case of neurotransmitters, there are some really important neurotransmitters that are not amino acids, monoamines, or peptides.
So this is going to be our acetylcholine category we talked about.
For example, this neurotransmitter right here is called acetylcholine. Acetylcholine. And acetylcholine is definitely one of our most important neurotransmitters. It does a number of functions in the central nervous system, and then in the peripheral nervous system, it's released by most neurons in the autonomic nervous system. Let me just write ANS for autonomic nervous system. And it's released by the neurons called motor neurons that synapse on skeletal muscle and tell our skeletal muscle to contract to make us move.
And on to features of neurotransmitters, where here we're investigating acetylcholine and taking an in-depth look at that specific neurotransmitter. So you can turn to your worksheet now, and we're going to work through this image here piece by piece. So I'm just showing you this slide to give you a broad overview, and then we'll actually work through this image. We'll break it down and look at each, like the synaptic knob and then the target cell. So we're going to look at an overview here, and that's going to include the synthesis and release of acetylcholine, the removal from the cleft, and then acetylcholine's target cell interaction. So here we're looking at the presynaptic neuron, and specifically we're looking at the synaptic knob here. So part A is going to involve the synthesis and release of acetylcholine. So you may have guessed the synthesis is going to involve the combination of acetate plus choline. So due to glycolysis that's occurring within the cell, we end up with pyruvic acid. And we can take an acetyl group off that pyruvic acid and through enzymatic activity combine it with choline to give us acetylcholine. Okay? And now that acetylcholine is going to be stored in vesicles within that knob. Okay. Then when we get an incoming nerve signal, we get opening of calcium channels, and we get an influx of calcium, allowing these vesicles then to fuse and dump their contents into the synaptic cleft. Okay. Now, thousands of molecules of acetylcholine are actually released by exocytosis into that cleft with that incoming nerve signal. And I should also point out that the more nerve signals we had, so the more signal, that equals more acetylcholine release. Part B, we're going to look at the subsequent removal of acetylcholine from the cleft. So actually, almost immediately, some of those acetylcholine molecules are going to be digested by another enzyme, acetylcholinesterase. Okay, so that's going to come along, break this back into choline and acetate, at which point the choline is going to be reabsorbed by that cell. Okay? So acetylcholinesterase is going to break down acetylcholine back into acetate and choline. And that choline is going to be reabsorbed. Okay. Now, however, some of that acetylcholine is going to move across that synaptic cleft and bind to receptors. But it does so only briefly, actually for about 1 millisecond before it drops off and then also is digested by acetylcholinesterase. Now let's look at the target cell interactions. So some of that acetylcholine is going to move across the synaptic cleft and bind receptors. And it can bind either nicotinic or muscarinic receptors. If it binds nicotinic, it binds directly. Okay. So here we see acetylcholine, and it's going to bind here to this nicotinic receptor, causing that ion channel to open in a direct fashion. Okay. In this case, that's going to lead to EPSPs, excitatory postsynaptic potentials. The binding to muscarinic receptors is indirect. Okay, so binding here. What's going to happen there is we're going to use a second messenger pathway involving some type of G protein. So we're going to activate this G protein due to the binding here. Second messenger pathway, and interestingly, this can cause an excitatory postsynaptic potential or an inhibitory postsynaptic potential. Okay. So how is it possible then for acetylcholine to generate either an EPSP or an IPSP by binding to a muscarinic receptor? Well, what I didn't mention yet is there are different subclasses of muscarinic receptors. So depending on the type of muscarinic receptor that we bind, we can get an EPSP or an IPSP. Okay. Now, I do have a
Think, pair, share for you here. So what I want you to think about is what could we do to increase the effects of a neurotransmitter? So take a minute and think about that. Well, you may be thinking about the action of antidepressants. So in a few slides, when we talk about antidepressants, some of you may have heard of like SSRIs. We're going to talk about how we can increase the time of that neurotransmitter within the synaptic cleft, allowing it to bind receptor, or maybe we'll talk about releasing more neurotransmitter into the synaptic cleft. So in that way, we could see an enhanced effect of that neurotransmitter.
Moving on to neuromodulation, and then we'll take an in-depth look at endocannabinoids.
Your central nervous
In addition to neurotransmitters, some neurons secrete a chemical called a neuromodulator. These chemical messengers, however, do not lead to the formation of EPSPs or IPSPs. Instead, they create more long-term changes, not like a neurotransmitter that is a very, very short time duration, right? And these long-term changes, they modulate the action of the synapse, hence modulation. Neuromodulators are also different from neurotransmitters in that they do not bind to receptors that are on the subsynaptic membrane, so the membrane that's right across that synaptic cleft. Now they can bind to both presynaptic and postsynaptic receptors, but they're just not found on that subsynaptic membrane. So if we have some neuromodulator that's been made by one of the neurons, that neuromodulator can bind to a neuromodulator receptor. And one of the things it can do is it could increase enzymatic activity within that neuron. And let's say that enzyme helps synthesize more neurotransmitter. Well, that over time is going to lead to more neurotransmitter that can potentially be released from that presynaptic neuron. Now, some other things neuromodulators do or don't do is they do not alter membrane permeability and potential. We kind of mentioned that in the previous slide. Another thing is the duration. These effects can last days to weeks to years, not the fractions of seconds we're talking about with neurotransmitter reception. Neuromodulators include neuropeptides. In fact, that's the largest class of neuromodulators. As well as ATP, adenosine triphosphate. So this normally is our energy carrier, but it also can be released into the synaptic cleft, and there it can move to bind some receptor on either presynaptic or postsynaptic neurons. Nitrous oxide, or NO, we'll talk about its role in vasodilation later. And then finally we have these endocannabinoids.
nervous system is
Endocannabinoids are neuromodulators that influence the same receptors as marijuana. So they're these small non-polar molecules that are made by your cells. Kind of think of it as endogenous cannabis. So they're made and released by postsynaptic neurons. So if we look on this illustration here, we'll see this little green key that's being synthesized. This is representing our endocannabinoids. And what they do is they go and they bind to a receptor on that presynaptic neuron. Okay? And they have effects on presynaptic neurotransmitter release. So binding of that endocannabinoid is going to decrease neurotransmitter release from the presynaptic neuron. And it has the same effects that we kind of associate with marijuana or being high. It changes your learning and your memory, and it affects your appetite. It also helps suppress nausea. And we will finish up today by looking at antidepressants, how they work, and then focusing on your mental health.
Pretty much comprised of your brain and your spinal cord. So the brain can be...
All right, so on your worksheet, you're going to want to fill out a brief mechanism of action for each of the antidepressants that is discussed within this short video.
And the antidepressants that we are going to be talking about today work on this synaptic level, as do many other medications. Because by increasing or decreasing the amount of available neurotransmitter in the synapse, medications can make it more or less likely that a message will be triggered in the next cell. And depression itself seems to work on this level. Studies have suggested that depression might be caused by low levels of certain neurotransmitters, called monoamine neurotransmitters.
Or your biogenic
And this includes serotonin, norepinephrine, epinephrine, and dopamine. And different theories disagree about the relative contributions of each one. But this is something I want you to keep in mind when we talk about how these medications work. Because all of the antidepressants we're going to cover in this video work by trying to correct this imbalance. They all cause an increase in the levels of these neurotransmitters, but they do it using very different mechanisms. So we're going to cover 3 classes of antidepressants: monoamine oxidase inhibitors, or MAOIs, tricyclic antidepressants, or TCAs, and selective serotonin reuptake inhibitors, or SSRIs. So let's start off with these MAOIs. And the clue to how these antidepressants work is in the name. Monoamine oxidase is an enzyme that breaks down neurotransmitters that isn't stored in vesicles. So it's kind of like cellular housekeeping. Monoamine oxidase inhibitors are drugs that inhibit the actions of monoamine oxidase. So by inhibiting this enzyme, it actually increases the amount of neurotransmitter that is capable of being released into the synapse. And more neurotransmitters in the synapse increases the likelihood that they will dock onto the postsynaptic cell and cause an action potential. The next type of antidepressants are tricyclic antidepressants. And instead of being named for what they do, these drugs are named for how they look. And I have a few examples here. And you can tell that all of them have this 3-ring structure. So tri for 3 and cyclic for the rings. And tricyclics work by increasing the levels of 2 specific neurotransmitters, norepinephrine and serotonin. But the way that they do this is very different from MAOIs. Instead of stopping an enzyme from breaking down these neurotransmitters, TCAs work by interfering with a very different housekeeping mechanism called reuptake. So much like you, your body likes to recycle. It likes to be efficient. And this is even true at the neuronal level. After the neurotransmitter has released into the synapse, your body doesn't get rid of it. Instead, it recycles it by taking it back up into the presynaptic cell through these reuptake channels. And then it can be repackaged into vesicles and released again. TCAs work by blocking these reuptake channels. And the result of this is that the neurotransmitter stays in the synapse longer, which then increases the likelihood that it will dock onto a receptor on the postsynaptic cell and trigger an action potential. SSRIs, or selective serotonin reuptake inhibitors, they work the same way. They work by blocking reuptake channels. But instead of blocking them for both norepinephrine and serotonin like TCAs, they only block reuptake for serotonin, and only for very specific serotonin receptors. So like MAOIs, here we have a situation where the name of the drug is a description of what the drug does. And I think of all of these antidepressants, SSRIs are probably the ones that you are most familiar with.
All right, so I'm not getting into it too much, but I should also say that the MAOIs and the tricyclics, they're kind of considered messy drugs or first-gen antidepressants, and they're generally turned to if SSRIs do not work for some reason. And it's because they're not very specific, and they can affect kind of the entire nervous system, whereas the selective serotonin reuptake inhibitors are more selective, so you get less side effects. Along with today's worksheet, I'm going to include this patient health questionnaire, or the PHQ-9. Now, I'm not doing this so you can kind of self-diagnose yourself and just say, hey, I have depression, but I want to give you some indication of where you are at during the semester. So you can kind of look at these questions. You'll see this if you go in for a checkup or some type of mental health check-in. They'll give you this, and then they look at the total, the sum of all of your responses. They'll add those up, and if you're above a certain value, you may be predisposed for dysthymia or depression. So I want to put this in there. If you get some time, you can take a look at it and kind of just do a check-in with yourself and see where you're at. I'm going to show you this Venn diagram here of how we arrive at mental health. So it's 3 components: biological, social, and psychological. So biology is going to include things like your genetic disposition towards maybe some type of mental health disorder. Maybe it includes your physical health. And then we have a social component. This can include major things like your family and your friends. And then the psychological component. How do you deal with different circumstances? And when all 3 of these come together, in the middle there, that's where we have our mental health.
Broken down into it.
And I'll finish up today just discussing kind of just having the blues versus depression, right? You've probably all been there where we're just kind of feeling down, but that's not always an indicator of depression. It's more long-term. So having the blues is normal and healthy after certain life events. Certainly if you had a grandparent that passed away, you're going to have the blues. Clinical depression is different. It's an illness caused by a chemical imbalance in the brain, as you just learned. Lasts for longer than 2 weeks, so there's a duration, and it leaves a person unable to function in just areas of their normal day lives, including your schoolwork, your social life with your friends, your job if you're working. And it is not a choice to have depression. It is a recognized illness with a biological cause.
A few different reasons.
Symptoms of this are going to include being depressed most of the day, losing interest in things that you used to enjoy. Perhaps you are on the soccer team and you don't get a kick out of it anymore. Terrible pun. You're seeing noticeable changes in your weight, changes in your sleep. The interesting one here, just your body motions tend to be in slow motion. You feel kind of like you're moving through a tunnel. And you obviously have fatigue. And then you have these feelings of worthlessness or hopelessness, like things are not going to get better. You have trouble concentrating, and that may include frequent thoughts of death. And we do have resources available for you if you are feeling down. I've included the phone numbers here. They're for both CSB and SJU, and I encourage you to make an appointment. I had one class when I was an undergrad, and the professor said, independent of our mental state, we needed to pass this class. In order to pass this class, we had to make an appointment with a mental health counselor. And we did it. It was great. It was a good thing to just sit down and, talk with someone about your mental health. These are great resources. I took advantage of them all the time in grad school. Don't be ashamed to take care of your mental health.
Regions. This highly folded area up here is called the cerebrum, while this central core right here is called the diencephalon. Then we have this here called the brainstem, with this really interesting cerebellum right behind it. But this brainstem is going to connect, you can see that we've cut it here, with the spinal cord. And that spinal cord is going to descend all the way down your back until it ends in that lumbar region. But essentially, if you're talking about the CNS or central nervous system, you're talking about your brain and your spinal cord.